Hello, this video lecture is going to accompany the current OpenStax microbiology textbook, pages 359 through 384. It will cover the sections 9.1, How Microbes Grow, and 9.2, Oxygen Requirements. The purpose of these sections is to familiarize yourself with 1. How bacteria in particular grow and divide, 2. Methods used in the lab to determine how much bacteria is present, and 3rd, how oxygen can affect bacterial growth. So let's begin by discussing how bacteria grow and divide. First of all, bacteria can only reproduce by means of asexual reproduction by the process known as binary fission. Binary fission is an asexual process that bacteria use to divide into two. First, their single chromosome has to be duplicated as shown here in step 2. The two chromosomes are usually attached to the inside surface of the cell membranes and are slowly pulled apart towards opposite poles of the cell, as shown in figure 3 or step 3. In the meantime, as the cell is growing in size, it will also increase its intracellular components, and eventually the cell membrane and the cell wall will form what's called a septum. This septum will internalize until two small daughter cells are pinched off and form two individual cells that are basically identical clones of each other. That's steps 4, 5, and 6. Notice that at all times there is no exchange of foreign DNA, that the DNA present in the original cell is duplicated and the daughter cells have identical DNA. So essentially the daughter cells are clones of each other. Now let's take a closer look at the process of the septum formation and the final cellular division. During this binary fission, the bacterial cells will make a protein called the FTSZ protein that will slowly assemble together and form a Z ring, as shown here in this slide. This ring acts as the tightening of a belt. It constricts the center of the cell. The Z ring determines where on the cell it will divide into two daughter cells. As the Z ring constricts, or as it tightens, it's going to form a septum. This is shown as the internalization of the cell membrane. Eventually the cell membrane meets together as the cell ring tightens all the way, and at the same time, the bacterial secretes enzymes that disrupt the peptidoglycan cell wall. This in effect breaks the covalent bonds found between the NAG and the NOM sugar subunits of the peptidoglycan. Other enzymes will help expand on these existing peptidoglycans to form a cell wall into the septum, or the septum grooves. So as the septum continues to grow, the cell membrane grows and the cell wall will grow with it. At the end, the two small daughter cells will have a complete cell membrane and a cell wall. These cells in turn will grow and continue to grow and expand and will continue the process of dividing in the same manner. So each time a cell divides into two, these two daughter cells then divide into two more cells as shown here. So the number of generations, the first generation, we start with one cell. That one cell led to two daughter cells in the first generation. Now these two daughter cells will continue and they will divide one more time, or a second time for the second generation, to form 4 daughter cells. These 4 cells then divide a third round, forming 8 cells, etc., etc. So in this manner, the number of cells continue to increase, what we call exponentially. So as this figure shows, in theory, 20 rounds of cellular division, or by binary fission, will result in over 1 million bacterial cells. If you recall seeing bacterial colonies on an agar plate in the lab, this colony started out by plating a single bacterial cell in the agar. The cells grew and multiplied by binary fission until roughly these 1 million cells crowded together to form that one visible colony. Now, some bacterial cells will grow and reproduce faster than others. Microbiologists have developed methods to study this rate at which the bacteria can divide and help them understand the progress of disease associated with these particular bacteria. So when studying bacterial growth, microbiologists like to know how long it takes for a bacterial cell to divide into its two daughter cells. This is often expressed as the generation time. The generation time is the interval or the time that it takes for one cell to divide into two more cells. But microbiologists usually study populations of bacteria. It's a lot easier than studying a single cell. So therefore, the generation time could be measuring the time that it takes for a number of cells within a population to double. This is also called the population doubling time. So the doubling time does vary among bacterial species, such that some multiply faster than others. Okay, so how do we do that? How do microbiologists measure this doubling time? We can measure the effects that certain conditions will have on bacterial growth by studying their growth patterns in the lab. These patterns take the form of a growth curve as shown here in this slide. Most bacteria growth curves are plotted using a closed culture method. That means that the bacterial cells will grow and divide with no extra nutrients added to the system and with the resulting metabolic waste remaining, so they are not removed. Now this slide shows what a typical growth curve for a bacteria growing in a closed culture would look like. Initially, the bacterial population would be low and they would not change. So during this so-called lag phase, as shown as number 1, the bacteria are just getting used to their environment. So whatever number of bacteria that we introduced into this closed system, that number is what we start out with. These bacteria are taking in the nutrients, they're metabolizing, they're making all the cellular components that they need to grow, but they've not yet divided. Now once they reach the point that they do begin to divide, the cell division will continue to progress exponentially. The faster that the cells divide, the shorter the doubling time is going to be presented. So this is reflective of the steepness of the line that is represented here as the log phase in number 2. So during the log phase, this is when cells are actively dividing and they're dividing exponentially. The steeper the slope will define the optimum growth conditions. Now, microbiologists can modify these conditions such as the amount of oxygen, the temperature that the cells are growing in, the presence or absence of certain nutrients, etc., etc. This will determine what the best conditions are for that particular population of bacteria to grow in, and will also affect the slope of that exponential growth. This information that they acquire is then used to understand the progress of disease or to optimally grow bacteria in a biotech industry setting. So for instance, if we can slow down the population doubling time in a person that's infected with a bacteria, we can, in essence, stop or reduce that disease prognosis. By stopping the number of bacteria, then hopefully the patient doesn't get disease or can recover a lot quicker. Now, these growth curves gives us a picture of what the bacteria needs to grow. Now, if the cells are allowed to continue to grow past this log phase, they'll eventually reach what we call the stationary phase, as shown here in number 3. At one point, the rate of cell division starts to slow down. This is due to the fact that we have a closed system. As the cells grow, they produce metabolic waste, which accumulates in the culture. They continue to feed on the nutrients, which are eventually depleted, and the oxygen levels are also depleted. So during the stationary phase, the number of cells created by cell division, or by binary fission, will equal the number of cells that are actually dying, so that growth curve will stay horizontally. Eventually, the number of dying cells will increase while the number of newly formed cells will decrease, thus lowering the total number of live cells. This is shown here as the death or the declining phase in number 4. Now, Outside the lab, bacteria grow essentially in an open culture with continual changes in its surroundings. We also have ways to examine bacteria in an open culture system. However, these are a little bit more complicated. As helpful as these growth curves are at providing information on the bacterial's nutritional needs, outside the lab, bacteria often grow alongside other genus or species of bacteria. In this natural setting, they often transfer their own DNA from one bacterial cell to another. Now, one way that a bacterial can pick up or transfer DNA is through the process known as conjugation. Now, if you remember from previous lectures, this method involves the formation of a sex pilus by the donor cell. The donor cell would be the one here on the left. The donor cell has a plasmid DNA that gives it the genes to form that pilus. Now, this plasmid DNA from the donor cell is duplicated and it's transferred via the pilus to the secondary or the recipient bacterial cell, the cell on the right, thus giving the recipient cell the additional DNA or genes needed to make this pilus. But oftentimes, that plasmid also contains additional genes. So this is a way of transferring DNA from one cell to another. Now, a second method of transferring DNA from one cell to another is by the process known as transduction. Transduction does not involve a pilus. In fact, it involves a virus that infects specifically bacteria. It's called a bacteriophage. The infected bacteria will make hundreds of new viruses before it dies. In the process of making these virus particles, its DNA will sometimes get packaged accidentally into the new viruses. These new viruses then go on and infect new bacterial cells, and they transfer their own DNA along with pieces of DNA from the previous bacteria, the donor bacteria that no longer exists. Thus, in transduction, it involves the transfer of DNA from one bacteria to another through the use of a bacteria virus. The last way in which DNA can be transferred is called the process of transformation. And this is simply just using pure DNA. So when a bacterial cell actually dies out in nature, they tend to burst open and release their DNA in small fragments. Some of the bacteria that are around in the vicinity of that dying bacteria are able to pick up or absorb these small pieces of foreign DNA across their cell membrane into their cytoplasm. Now, if these pieces of DNA are similar enough to their own, they can take them up inside their chromosome and thus acquire additional genes. So while DNA is mostly transferred from mother to daughter, we sometimes call this vertical transmission, they will tend to form clones. There are times when bacteria can actually pick up and express additional DNA from other bacterial cells. This is called horizontal transfer. Thus, this helps to strengthen or could also, in effect, harm the population. So it could go either way. But what we should know is that this is an important issue when we start to study the spread of antibiotic resistance or the spread of toxin genes from one bacterial species to another. So far we've talked about growth curves, the need to measure the presence of bacteria, and I've also just digressed a little bit and talked about the transfer of DNA during those growth processes. So when do we need to know how many bacteria are present? Well, the first one we need to know to study their growth curves to see how fast they grow and what nutrients are needed. Another reason why we need to know how many bacteria are present is mostly for clinical microbiologists. So clinical microbiologists often need to know the number of cells present in a clinical sample. So for example, bacterial counts of greater than 100,000 bacterial cells or colony-forming units per 1 ml is very indicative of an infection, specifically if it's a urinary tract infection. We also need to know how many bacterial cells are present, say, in our drinking water. So water reservoirs are highly tested. They're constantly being monitored for their bacterial counts as any indication for possible microbiological contamination. I'll come back to how we monitor our drinking water in a minute. And lastly, a final reason of why we'd want to know about the number of bacteria that's present would be in our food. So food products are often tested along the various processing steps during the food industry processing steps to help improve the quality of the food and extend its shelf life. So for example, if that food has low bacterial counts, it will remain fresher longer and can allow the seller more time to sell their products. At the same time, the food is fresher once we receive it and when we actually consume it. So these are 4 reasons of why we'd need to know about bacterial counts that are mentioned in this chapter. Okay, the methods that are used to determine the number of bacterial cells can be divided into either direct methods or indirect methods. Now they're divided basically on the fact that direct methods actually count the number of individual cells. They do that directly. Whereas the indirect methods do not physically count the number of cells. They rely on an indirect way. Makes sense. Okay, so there's going to be 3 direct methods that are listed in this chapter. And the first one requires a Petroff-Hauser chamber. Now this chamber is shown here in this slide, and it looks very similar to a hemocytometer that you might have seen that's often used in the hospitals or the clinical settings to count red blood cells. So this chamber, shown in the middle figure, basically will have a cover slide that's associated with it. This glass cover slide is placed on top of the chamber, and a bacterial suspension is added just underneath the cover slide. It's going to fill the chamber, and the area and volume that the chamber contains is predetermined. So for example, showing to the right here, we're blowing up a small etched figure. So in that chamber, there's a mirror type of finish that is etched with lines. These lines will form these squares as shown here on the right. This particular square, shown in a red circle all the way to the right, has an area of 0.008 millimeters cubed, which actually is equivalent to the volume of 0.00001 mil. Now in that square there are 10 cells that are labeled or indicated. So the number of bacteria in this sample is said to be 10 cells per 0.000008 mils. This is then converted to number of cells per 1 mil. Okay, so let's hope I got the number of zeros correct. If you want to pause this video and look at this slide, I've highlighted basically what I just said in yellow. Now usually when a clinical microbiologist counts these cells, they would count the number of cells in at least 3 or more squares and take their average to help increase their accuracy. Now however, this method does include both the dead and the live cells as their total count, their total cell count, because we can't distinguish using this method as presented to knowing whether the cells are alive or dead. Now, there are times that we would only want to know the number of dead cells or the number of live cells. So in this case, a differential staining technique called the viability staining or the live dead staining can be used alongside with a fluorescent microscope. And that's shown here in this slide. So in this slide, the cells were stained using 2 stains. The first, or the primary stain, is a green fluorescence dye that will enter all the cells that have an intact cell membrane. So therefore, the green will stain both the live and the dead cells. These cells are then washed and next treated with a secondary stain, which is a red fluorescent stain, which can only enter the cells if the membranes have been damaged. So the red dye will only enter cells that are dead. So as shown in this figure, we have the majority of the cells are green. These are live cells, and there are a few cells that are red. These are the dead cells. So again, we could count within a certain area and calculate the number of cells per mil that are either live or dead. Now the second direct method of determining number of cells includes the viable plate count. The viable plate count method can be used to calculate the number of live cells only. So in this method, only the viable or live cells can grow and replicate on agar plates that have the necessary nutrient conditions for those cells to grow, and thus form colonies. Now there's two limitations to this method. The first one is that if the single bacterial cells are plated too close to each other and grow too close to each other on the agar plate, they will give out a single colony forming unit. So two bacterial cells will appear as one colony. A second limitation is that if the bacteria that's being counted grows in chains, such as streptobacilli or streptococci, or in clusters, such as staphylococci, they too will grow in too close to each other and appear as a single colony. So knowing these limitations, this technique is still very useful and is used to estimate a low is used to give a low estimate of the actual number of live cells. The reason why most scientists use this method is because it is easy to perform and doesn't use expensive equipment. Now, oftentimes to set up a viable count experiment, we first need to do a series of dilutions. That's because the sample that contains the bacteria might be highly concentrated. That means it might have too much bacteria. So we need to dilute it so that when it comes time to plate the bacteria, that we reduce the chances that they will grow into each other or close to each other to form a single colony. So in this case, serial dilutions is the first step to help ensure that we would put on a plate anywhere between 30 to 300 colonies, bacterial cells, on an agar plate. Sometimes we call bacterial cells colony forming units for the simple fact that a bacterial cell is the result, forms actually a colony. Sometimes we will call it CFUs. CFUs is an acronym for colony forming units. I just realized that there's a mistake on my slide. Instead of unites, it should be units. Okay, so these series of dilutions will prevent overcrowding of the cells, or equally damaging is having too few cells to count. Either way, getting colonies between 30 and 300 will greatly reduce the errors and increase the accuracy of calculating the total number of live cells. Okay, so in this slide, I'm showing you a sample, an original sample shown in this bottle, and we're going to remove 1 ml of this original bacterial culture and add it to a tube that has 9 ml of sterile liquid. In effect, what we're doing is diluting the original sample 10 times. We're adding 1 ml of bacterial sample to 9 ml of sterile solution. So we've diluted this sample 10 times. We can represent this as a 1 to 10 dilution. This newly diluted sample is then diluted again 10 more times, again by taking 1 ml from this diluted culture and adding it to a second tube that has 9 ml of sterile water or sterile liquid. This process is repeated 3 more times, and a small amount of each of the diluted samples is then plated on an agar plate and allowed to grow. Now the number of bacterial colonies or the number of colony forming units are then counted and used to calculate the number of live bacteria that's present in the original culture. So the only reason to do these serial dilutions is if you start out with a solution or a sample that you know will have a lot of bacteria in it. But sometimes when we can perform viable plate counts on samples that also may contain few bacterial cells. So it's the opposite of having too many. We might have too few bacterial cells. For this, microbiologists need to basically use the membrane filtration technique. Now, an example of why a microbiologist would use this is that microbiologists routinely test drinking water reservoirs. So we're getting back to the water reservoirs that I mentioned earlier. So we would test these reservoirs for the presence of possible contamination. So in this case, a large volume of water is first filtered using a porous membrane to concentrate the number of bacteria that are present in the water. So as the water passes through the filter, the bacteria become trapped into this filter, and the membrane that this filter is made out of is then layered on top of an agar plate. The bacteria are then allowed to grow, and the number of live bacteria or colony-forming units are then again used to calculate the total number of live cells. So either way, viable plate counts. If the sample is too concentrated, we would first dilute them down using serial dilutions, or if there are too few, we would concentrate them by filtering the liquid through the membrane filtration technique. Okay, so the last direct method presented in this chapter is called the most probable number. Now this method is routinely used to test the number of coliforms that are present, again, in water reservoirs. So whereas the previous viable plating counts measured the total number of live bacterials present, the most probable number method will measure the total number of live coliform bacteria that are present. Now what's the difference? The difference is being the word coliforms. Coliforms are gram-negative bacteria that are present in the stools of humans and animals. So they're going to be used as an indicator of fecal waste. So an increase in coliform counts is indicated of the contamination of the water reservoir with fecal matter. Okay, so we're going to bank on the fact that coliforms can ferment the sugar lactose. When they ferment lactose, they form an acid as a waste product. So the media that we're going to use here will be a liquid broth that will use lactose as its sole carbon source and will include a pH color indicator. This pH indicator is red when the media is at a neutral pH such as 7, and it will slowly turn yellow as the conditions in that media turn acidic. So we're going to look for a color change from red to yellow. So as I'm showing you in this figure, there are 3 amounts of water samples that are being tested. Now, when we're all said and done, there's 15 tubes here. All 15 tubes will have the same amount of total volume. Okay, so 10 mils of water sample will be added to a first set of tubes. This set will contain 5 tubes. So all 5 tubes will be identical. They will have a certain volume of liquid media that has lactose and the pH indicator, and to those same 5 tubes, we will take 10 mils of our water reservoir. Okay, next, we'll add 1 mil of water sample to a second set of 5 tubes, and again, the volume will be brought up to equal the first set, but in this case, only 1 mil instead of 10 mils of water that we're testing will be added to these tubes. And lastly, the third set will contain 0.1 mil of the water samples. Okay, when all this is done, each tube, as I mentioned, will have the same volume, and the 15 tubes will then be incubated and examined within 16 to 24 hours later for the presence of lactose fermentation, as being indicated by the presence of a yellow color. Okay, these 3 sets of 5 tubes each will remain separate, and the total number of yellow tubes within each group will be recorded. So in this figure, all 5 tubes that received the 10 mils of water turned yellow after being incubated at 37 degrees for 24 hours, whereas only 2 of the 5 tubes that received 1 mil of water turned yellow, and none of the tubes that received 0.1 mils of water became yellow. Now these results are going to be used to match up against a table that's labeled as the most probable number table. Now a lot of work has gone into making this table, in such that repeated numbers of water samples with known amounts of bacteria were used to calculate or to get various results. So for example, our tube showed that the set of tubes, the 5 tubes that received 10 mils, all 5 turned yellow. Whereas the 5 set of tubes that received 1 mil of water sample, only 2 of the 5 fermented lactose and turned yellow, and none of the tubes that received 0.1 mil of water changed, meaning that there was no bacteria present in those tubes. Now, based on all the work that went through to create this table, the statistical value shows that most likely there are approximately 49 bacterial cells per 100 mils in that water sample, based on these results alone. There's a couple other columns that I omitted that leads to its statistical importance, but basically what I want you to know without getting boggled down is that by looking at the number of tubes that turn yellow, we can estimate the total number of bacteria that are present in 100 mils of liquid. Okay, so let's jump. That's it for the direct methods. Let's go into the indirect methods now. The indirect methods of determining the number of bacterial cells mentioned in this chapter will use a spectrophotometer. Okay, what is a spectrophotometer? It's shown here on the left, and it's a machine that is equipped with a light bulb that has a system of filters that allows certain wavelengths to pass through a test tube that contains liquid that may or may not have bacterial cells. The amount of light that can pass through this tube is then measured by a detector. So obviously the minute we start introducing cells into the liquid, some of that light will be refracted, it will bend and not make it to the detector. So the cells in the liquid will interact with the light, preventing it from passing through and reacting with this detector. Goes hand in hand with the more cells that are present, the more visibly cloudy or turbid the tube appears, the less the light can penetrate through, therefore the less light is measured by the detector. The number of cells can be measured as a percentage of transmission, percentage of the light that actually penetrated through the tube, or it could be recalculated using the logarithmic value of the amount of light that was absorbed by the cells that did not make it to the detector. So either way, we can represent this as a percent of transmission or as an absorbance value. Ultimately, the more cells that are present in the tube, the lower the transmission value becomes. Now, it's also possible to correlate this turbidity or this transmission value into actual number of cells by performing a viable plate count with these samples, and then we could then associate a transmission value with that of total number of cells. Okay, so this chapter also talks about the oxygen requirements for bacterial growth. So one factor that can affect the ability to grow bacteria in the lab is the specific amount of oxygen that's needed by these bacterial cells to grow. Now, some bacteria can only grow in the presence or only grow in the absence of oxygen. Others will only tolerate small amounts, so they only need small amounts to grow. Whereas some other bacteria can grow both in the presence and the absence of oxygen. So microbiologists need to know this, because with the wrong amount of oxygen, then we won't get any bacterial growth, and we will not be able to measure the number of bacteria present in any samples. Okay, so to do this, the microbiologists are going to use a certain tube called the thioglycolate tube to determine which category the bacterial cells that they are testing will fall into. And this picture shows 5 such categories. So in this technique, the bacteria are going to be added to these tubes that contain the thioglycolate media. Originally, these tubes are depleted of oxygen, so they don't have any oxygen in them whatsoever. We're going to inoculate them with bacteria, and over time, oxygen will slowly diffuse throughout the tube, starting at the top of the tube. The area of the tube that has the greatest bacterial density, or its optimum bacterial growth, will occur when the right amount of oxygen is met. So as shown here in tube A, all the growth is seen at the very top of the tube. These bacteria are what we call obligate or strict aerobes. Obligate aerobes because they can only grow in the presence of oxygen. They cannot grow without an abundance amount of oxygen. So a little bit of oxygen, they still can't grow. Next, in tube B, these bacteria grew at the very bottom of the tube. These are obligate anaerobes, which means that any amount of oxygen will kill them. So these are tubes A and B are the two extremes. In tube C, there's more growth at the top of the tube and continuous growth throughout the tube after that. We're going to call these facultative anaerobes. Facultative anaerobes are organisms that thrive in the presence of oxygen, but can also grow in its absence by relying on fermentation or anaerobic cellular respiration. If there's any suitable electron acceptor that's there besides oxygen, the organism will be able to grow and perform anaerobic respiration. So the word facultative means it can grow with or without something. Facultative anaerobes means it can grow with or without the presence of oxygen. In tube D, there's growth that's evenly distributed throughout the tube. These are going to be aerotolerant. It's going to be difficult to distinguish between facultative anaerobes and aerotolerance, but the difference is that we have more growth on the top of tube C, whereas the growth in tube D are equally distributed. So these aerotolerant anaerobes in tube D do not use oxygen because they usually have a fermentative metabolism, but they're not harmed by the presence of oxygen. So they're obligate anaerobes, but the presence of oxygen does not harm them. The last one is tube E. In this case, the oxygen level has to be just right for growth to occur, not too much and not too little. So these microaerophilic bacteria require a minimum level of oxygen for growth, about 1 to 10%, well below the normal 21% that's found in our atmosphere. So they require small amounts of oxygen to grow, whereas the lack of oxygen, they cannot grow, and too much oxygen will kill them. Okay. Knowing where our bacteria falls is needed by specific bacteria will help us understand how they grow and to grow them so that we can study them. So I have a couple of examples here that the textbook provides. The first one they call them bacteroides. These are typically found in our intestines. Normally they grow symbiotically with us. They help break down our food, so ultimately they help provide us with various nutrients. However, when they're found in other parts of the body besides our intestines, they can still grow in the absence of oxygen, but they are the causes of abscesses and other infections. So given the right location, they're good. If they veer off to other parts of our body, then they can become pathogenic. Next, there are various species of Clostridium. The textbook gives you the example of Clostridium difficile. Difficile is often a secondary infection. Usually other bacteria prevent Clostridium difficile from attaching. However, in the presence of oxygen, they exist in the form of endospores. So endospores, they're not really actively growing, but the minute they enter our body and are able to attach, if we've disrupted our normal flora as an example, if they go into our intestines where there is no oxygen, where it's anaerobic, they can then germinate to form vegetative bacterial cells. Now in this case, they are a causative agent or the cause of inflammation of the colon. Okay, so this concludes the information that is covered in the textbook in sections 9.1 and 9.2. I will follow up with another video that will cover the remaining sections of Chapter 9.